All right, so last time, let me put into the screen so you can see it, what we're going to say, all right? Let me application screen share with you, and you can see what I'm going to project here, right? So the screen says like this, okay? So this is the last slide, if you recall. Everybody okay, right? If you have any question, let me know. And we did the polypeptide, remember? So A1 plus A2 plus A3, and you know the chemical reactions, right? So hydroxyl group from the carboxylic acid of the first amino acid, and H of the NH2 group from the second amino acid will make the dehydration reactions. Likewise, carboxylic acid from the hydroxyl group of the carboxylic acid from the second amino acid, and HN from the third amino acid will make a dehydration reaction, and it continues, same reaction, it continues like this way. And so the polypeptides are named when n equal 2, for example, only 2 polypeptide called dipeptide, 3 tripeptide, 4 tetra, 5 pentapeptide, and so on and so forth. Once we go greater than n equal 20, we call oligopeptide. All right, so greater than 20 or so amino acid polymerized, called oligopeptide, and greater than 50 we call proteins. All right, and I'll give you some example of the protein. One of the examples that I want to give you is the dipeptide. Dipeptide is aspartame, ERM or aspartame, right? It's a sugar substitute. Aspartame is sugar substitute. What does it have? It's a dipeptide. So it's a ASP. ASP stands for aspartic acid, right? ASP, L-aspa, and L, remember their L configuration. That's L stands for configuration of the aspa linked to the peptide bond with a phenylalanine, PHE, phenylalanine. All right. So this is the aspartame. So aspartame is a dipeptide, combination of 2 amino acids, L-aspa, L configuration, L-phenylalanine. If you have any combination of, so this is the sweeter, right? That's basically sweeter. About so sweeter, in fact, if you compare the sweetness, so it is sweeter than sugar, table sugar. So sweeter than sucrose. That's basically what you're saying, right? That's our table sugar. Then sucrose, much sugar, in fact, it is about greater than 200 times, sweeter, greater than 200 times sweeter than sugar. All right. Now, if you take any combination, let's say if you take a D, you know the configuration. So D-aspa, right? We all know the configuration. So if you take a D-aspa and L-phenylalanine, if you have like this, or for example, let's say you have a L-aspa, L-aspartic acid, any kind of combination, L-aspa and D-phenylalanine, so different configuration, D-phenylalanine, or if you say D-aspa, aspartic acid, and make a peptide bond. This is a peptide bond, Peptide bond with the D-phenylalanine. If you have any combination of this configuration, any combination, all three of them, they are bitter in taste. They are bitter. So think about it, how the configuration matters in this case, right? They are bitter in taste. They are bitter. So this is sweeter, and that's a bitter. So our tongue has been adjusted with this configuration alone, and therefore the configuration of the amino acids means a lot when they combine. Always L, right? Now, so as I said, greater than, So greater than, we have a 50 and proteins, 50 of the proteins, right? And examples are the enormous details. Example of the proteins would be, yes, hemoglobin, right? HB stands for hemoglobin.
So you're pretty much saying is that when it's 20, is more than 20 amino acids, it's that name with oligopeptide?
That is correct.
Okay, thank you.
And if it greater than 50 is a protein. An example of proteins, there are many. Hemoglobin. Can someone tell me what's the hemoglobin? What's the function of hemoglobin? Function of hemoglobin? I'm not going to give you all, but I'm going to give you some.
It transports oxygen in the blood.
Very good. Yes. Yes. So it is a oxygen carrier. So this one is a carrier protein. It's a carrier protein. So it is a oxygen carrier. Please write down. So basically hemoglobin is in the blood, red blood cells, and it binds to oxygen and it transports to all over the body. All the cells get the oxygen from hemoglobin. How about the myoglobin? Who can tell me what's the function of myoglobin? Myoglobin? Let's say MB, hemoglobin HB, myoglobin MB. What's the function of a myoglobin? Hemoglobin, myoglobin are causing. Yes?
Oxygen storage.
Oxygen storage in
The muscles.
muscles. Thank you. So oxygen storage. So oxygen storage in muscles. Please write down. This is a very important function. Likewise, GLUT. What's the function of GLUT? GLU, glutamic, GLUT, GLU is a glucose. GLUT, capital T. What's the glucose? It's a GLC, glucose transporter. Capital T is a transporter. Remember the channels I gave you already, right? So basically a transporter, all right? Transporter. There are many others there. I just give you some example, not too many, but at least you should know some function, right? So hemoglobin, oxygen carrier to all over the cells. Without oxygen, without hemoglobin, you cannot survive. Myoglobin is a storage oxygen in the muscles, and GLUT is a glucose transporter. I'm going to put etc., right? Etc. Glucose is the membrane protein because it is bound in the membrane. Remember the glucose, the carrier that we talked about it, right? The glucose is the neutral channel, and all those neutral molecules, sugar, glucose, fructose, mannose that can pass it through. It's a glucose. This channel, this transporter transport glucose. Similarly, you have a fructose transporter, you have a galactose transporter, mannose transporter, etc., etc. All those transporters are different. Those are channels or tunnels, neutral channels. All right, so that's basically it is. And there is another type of proteins. Example of proteins are called enzymes. Enzymes are vital to our life. Enzymes. All right. Now, when these proteins are synthesized, amino acids are polymerized to make the proteins greater than 50 or so, then they must undergo structural configuration. So the structure of the proteins. So it starts with a amino acid configuration. All right. So let's say AA1. I said say A1, but you can put AA1. Fine. AA1, AA2. Right. To the peptide bond attached to the peptide bond.
Professor Gardner?
Yes.
Do you think you could go back for 1 second?
Sure. All right. So yes. So hemoglobin, oxygen transporter, myoglobin, oxygen storage, GLUT, GLU, capital T, glucose transporter, and enzymes. There are many of them. I'm put etc. right here. There are many. We have thousands of proteins and enzymes. So enzymes, and I'll give you some glimpse of the enzymes. Okay. All right. Shall I go on? Move on. Okay. So if you have a amino acid 1, amino acid 2, and they are linked like this way, right? And ultimately, let's say amino acid AN. So that's a polypeptide. And let's assume N is greater than 100. I'm just giving a number, 100 or so. So when you have a amino acid are combined together and make a polymer of proteins, this is called primary structure or 1-degree structure, primary structure of the protein. And this is the non-functional. So primary structure of the protein is non-functional. Everyone okay? Then this has enough information to fold. So basically folding of those primary sequence folding of protein, and that will make a structure looks like this. You'll have it like this. Please write down with me. I'm not going to give you all of those, but some at least, right? You have like this kind of structure, like this, and you can also have a let's say another for an example like this, and then like this way, and like this way. All right. And the first one, if you look at very carefully, this one starts with the NH2. The amino acid, first amino acid has a NH2 free, and the last one is always carboxylic acid, COOH. And rest of the NH and carboxylic group acids are gone. They make a peptide bond. So this is the secondary structure. Secondary structure. So primary structure and secondary structure. In the secondary structure, you can see some sort of helix, right? This is a helical strand. This is called alpha helix. Yes?
My computer just now let me in, and I wanted to make sure you didn't say my name for attendance.
Say it again.
My computer just now let me into class, and I wanted to make sure you didn't say my name for attendance.
You're fine. You're fine, okay? You're fine. All right. So, and when you have so this is called alpha helix. So as this one, so as this one. And then this kind of structure that you see, one like this way, one like this way, these are called beta sheets. So this is alpha helix. These are beta sheets or called beta pleated sheet, but in general it's called beta sheets, beta sheet structure. All right. And then also have alpha helix like this side too. So, and they're connected with the beta turn. They're connected. Let's say, for example, the two beta sheets are connected with a beta turn. Beta, that's the beta turn. So secondary structures, beta turn. All right, beta turn. So secondary structures are three different types: alpha helices, beta sheets, and beta turn. But this is also non-functional. So let's say I'm going to put a NF, stands for non-functional, meaning it doesn't function. You know, the protein has a function, right? Okay, for example, hemoglobin binds to oxygen, glucose transporter, glucose transport glucose, but primary sequence doesn't do anything. So the secondary structure, it is also non-functional, no functional yet. So what happened, in order to have a functional, this molecule will fold again. This will be again folding. And in this type of folding, basically folding via weak interactions. Who can tell me what the weak interactions? Does anyone know what are the weak interactions? We talked about it. Weak interactions. Does anyone know what are the weak interactions? Yeah? In covalent molecules, what are the weak interactions? Give me one example at least. Yes?
Hydrogen bonds.
Thank you. So H bonds, I'm going to write for you. Give me another one if you can, because you already know. Yeah, give me another one.
London dispersion force.
Yeah, LDF. Why not? Give me another one. Yeah. So hydrogen bond, yes?
Dipole.
Yes, I'm going to write it for you. So you have ionic interactions, II and DD. Give me one more. That's the last one. DD, dipole, London dispersion force, dipole, ionic interactions, II, ionic interaction, hydrogen bonds, and give me big one. That's another important one. Give me HP. What's the HP? Yeah, hydrophobic interactions. And once you put it like this secondary structure will be converted into a very tiny particle. So you won't be able to tell. All right? It will be like this way. And N to C. So it's that's a three-dimensional structure, 3D structure. And this is functional. This is functional molecule. All right. So based on the function, you already gave me one such function actually. Hemoglobin is the oxygen carrier. Myoglobin is storage oxygen, right? It's stored oxygen. In the myoglobin, what you have inside, there's iron there. So myoglobin, hemoglobin and myoglobin both, they have a hemoglobin. So hemoglobin basically what happened. So for example, hemoglobin Hb, it binds to the oxygen, and this oxygen is bound to the iron. It's bound to the iron. So you'll have it like this. First one, it binds to one molecule of oxygen, O2, inside the protein, and then another 3 more molecules of oxygen will bind to it. All right. So plus O2, plus O2, plus O2. So basically, and plus O2. So in fact, and then it become a Hb. It binds 4 molecules of oxygen, and it goes to circulation. Remember, this oxygenation takes place inside the lungs, right? Inside the lungs. So that then it goes to the circulation. So hemoglobin has 2 different types of protein. One is called alpha protein. It's a name. Beta protein. Remember the ATP synthase will give the alpha, beta, gamma, something like that. Similarly, it's a subunit. So it is alpha 2, beta 2. Alpha 2, beta 2 has a 2 alpha polypeptide, 2 beta polypeptide. Each polypeptide binds 1 oxygen each. So that's basically hemoglobin O24, and it goes to circulations, right? All right. So that's basically our, and it is basically what happened here. In this, it is an iron inside. It is iron 2 plus, and that basically binds to oxygen. As it binds to oxygen, it is converted to Fe3 plus. Please remember, because I asked the question very routinely. Fe3 plus. In hemoglobin, first thing first, what's the function of hemoglobin? It's oxygen carrier. It binds to oxygen and it transports to all over the cells. Number 1. Number 2 questions I ask always, that how does it bind to oxygen? Basically what happened, iron hemoglobin, which is not oxygenated, called deoxygenated hemoglobin, and that's why you see the blue color oxygen, a blue color in the vein, right? In the vein there's a type of blood called deoxygenated blood. All right, that's one carrying that one doesn't have the oxygen bound to it. So it is Fe2 plus. Once it binds to oxygen, it is oxidized, and now it is binding to, it becomes like this way, and it is Fe3 plus. So Fe3 plus binds to oxygen. It goes inside it goes on the cell surface, delivers the oxygen, become Fe2 plus again, and brings it back to the vein, to the lungs, gets the oxygen again, and goes again and again. It's a circulation. All right. The next major type of proteins is enzymes. Does anyone have a question about the function of hemoglobin? I'm not going to go to details, many of those. So yes?
I had a question on the very top one where you have, I can't tell, where you wrote folding. I don't know what that next one is that you wrote.
This one?
The one over from that.
So A1, A2, A3.
You have it. It's the one underlined right below you.
Right below, right here?
Yes.
Yeah, it's a beta turn, T-U-R-N, beta turn.
Okay, thank you.
You're welcome. Alpha helix, beta sheets, beta turn. And now these are functions. So hemoglobin is a transporter protein. It transports something. Likewise, we have a LDL. You know the LDL, yes?
I just had a quick question. So when you said hemoglobin has alpha 2 and beta 2, do those come from the alpha helixes and the beta sheets?
No. Very good point. Those alpha and beta, the alpha and beta is the name of the polypeptide.
Okay.
Yeah. Remember that ATP synthase has A, B2, C8 to 10, 8 to 15, and there's a F1 has alpha 3, beta 3, gamma, delta, eta, the Greek letters. And so this is the alpha 2, beta 2 is a heterotetramer. It is a heterotetramer, 4 polypeptide together. And within the polypeptide, there are plenty of alpha helices, beta sheets, many of them. Many of them. Yeah,
okay,
yeah.
And so the alpha and betas within hemoglobin just hold it together?
Yes.
Okay,
Yes.
and
That's
do they
right.
have any other function in carrying oxygen other than holding hemoglobin together?
No, only thing, the alpha 2, beta 2 fold together such that the 4 polypeptide are together. They can bind 4 molecules of oxygen. So
Okay.
it's very, very economical. So you can transport 4 oxygen molecules with 1 molecule of hemoglobin to all over the cells. Yeah. All right. So the next major class of proteins is the enzymes. And enzymes are the catalyst. Enzymes are catalyst. Remember, we have without enzymes we cannot function. Right? Enzymes are catalyst. So catalysts are catalyst. Enzymes are catalyst. So the catalyst. So the important properties of catalysts, they do not change the structure, 3-dimensional structure, before and after. Before means before the binding to the substrate, converted into product, and structure remains same. Please write down. So no structural change. Number one, no structural change. No structural change during the biochemical reactions. No structural change. All right, no structural change. No structure. All right. Structural change. No structural change. Please write down. So no structural change before the reaction, after the reaction. That's number one. Number two properties, basically, it has the no structural change, and it lowers the activation energy. So any chemical reaction, they need some energy input. We call activation energy. So catalyst, basically they lower the activation energy. Activation energy is the energy required to start the reaction. So activation, let me write down first. So lower the activation energy. Lower the activation energy. Please write down. These are properties of the catalyst, and enzymes are catalyst. All right. So lower the activation energy. All right. So no structural change before and after the reaction. Lower the activation energy, and number three, speed up the reaction. All right. So speed up the reaction. Please write down. These are the properties of the enzymes, catalyst. Speed up the biochemical reaction. So speed up the reaction. All right. So these are the 3 different criteria, and you must remember. And that's basically to speed up the reaction. If you have any question, please ask. So enzymes are catalyst. They bind to the substrate to convert into product. And during that, no structural change before the reaction, after the reaction. They remains as it is. They lower the activation energy to speed up the reaction. Activation energy is energy required to start the chemical reactions. All chemical reactions need activation energy, either low or high, that depends on the reaction, but they need some. The enzymes reactions are very fast, therefore they lower the activation energy such that those reactions do not need too much energy input. A little bit energy is good enough to carry out the reaction, to speed up the reaction. And as they lower the activation energy, the enzymes speed up the reactions. These are properties of the enzymes. As I say, enzymes are catalyst, right? All enzymes have 3 different properties, 3 different unique characteristics. One is they have a catalytic power, enzyme specificity, and enzyme regulations. What's the catalytic power? It is also called CP. C for catalytic, P for power. So catalytic power equals, all right. So I can write CP equals, just write down the definition, good enough. Is a ratio of rate of the reaction all right, rate of the reaction with catalyst, with enzyme, rate of the reaction with enzyme divided by rate of the reaction Ditto. Ditto means the same thing. Rate of the reaction without the enzyme. So with rate of the reaction with the enzyme, rate of the reaction without the enzyme. Please write down, okay? Without the enzyme. So that's basically the rate. And they have a very high CP, the catalytic power. And for example, many enzymes can have catalytic power from 10 to the power 6 to 10 to the power 28 order of magnitude. Very, very high speed, high catalytic power. All enzymes can perform at a very, very high speed. Very, very high speed. And that defines the catalytic power. Catalytic power is the rate of the reaction with the enzyme and divided by rate of the reaction without the enzyme. An example is like this. Enzymes also have a specificity, meaning one enzyme, specificity means one enzyme, all right, one enzyme binds only one substrate, one type of substrate. So one enzyme binds only, it can only one type of substrate. That's a specificity, type of substrate. I'll give you an example within a minute. So substrate, all right, substrate. So let's assume, so one type of substrate, that's a specificity. For example, let's say we have an enzyme and three-dimensional structure looks like this, all right, and here you are going to answer to me like this. And so this is the enzyme we call E, all right, and this cavity that you see the hole, that's a substrate binding site. So let's assume we have a different type of substrate. So a substrate looks like this, another substrate looks like this, another substrate looks like this, all right, I'm going to put it like this. Another substrate looks like like this, etc. Let's put another substrate. Another substrate looks like this. So these are all substrate. Substrate 1, substrate 2, substrate 3. Let's say this is our S3, for example. S3, S1, S is the substrate, S2, S4, 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 and S5. Just you could have many. Now, who can tell me which one will bind to the enzyme? Among these substrate, which one will bind to the enzyme? Tell me. I'm going to wait for your answer.
S3.
Does everybody agree? And in fact, it is. That's right. So basically, S3 will bind to like this way. Correct. Because it has the same type of configuration like this. So this is a S3. And therefore, it will make a ES3 complex. It will never, this enzyme will never bind to the S1, S2, S4, S5 because they will not fit into the substrate binding pocket. This is called specificity. One enzyme binds only one type of substrate and that's it. All right. Enzyme regulation. This enzyme binding and ultimately what will happen, this will go to enzyme again. Let's say there's E. I'm writing E and plus P. P is the product. So you have E plus S, ES complex, and then ultimately enzyme will retain and it will make the product. So chemical reaction will take place like this. And how fast? It could be 10 to the 6 order of magnitude to 10 to the 20 order of magnitude. It's very fast, exceedingly fast. This activity, enzyme activity, is regulated by many different factors. And those factors, so enzyme regulations, there are several factors are there. All right, I'm going to write only few. There are many factors are there. So factors that regulates means the turning off and on. So the factors are, there are many factors are there. I'm going to give you only a few. the temperature, the pH of the buffer of the media or inside the cell. Remember, we have two types of buffer, right? Closed system, open system. The pH can change the rate of the reaction with enzyme, because the pH can change if you at all enzymes specifically perform at a particular pH. For example, we have some enzyme called pepsin, and that is present inside our stomach, and that enzyme works in a very low pH, pH 0.8 to 1.8, pH 1 to 2, within this range it works. Why? Because our stomach has plenty of acidic, HCl. Whereas all the enzymes that we talked about during the glycolysis, TCA cycle, electron transport chain, they all works at 7.4, because that's our physiological pH. So every enzyme has a specific pH. Their activity is optimum in a particular pH. So pH is one of the factors that can affect the change of the activity of the enzymes. B is a salt concentration. Salt concentration are basically our inside the cell. We have a potassium chloride about 100 millimolar, and outside and sodium is about 5 to 10 millimolar. Low sodium, high potassium, and that's basically we need. If that potassium concentration goes lower, many of the enzymes do not work, and that's how you get the cramp. Remember, right? So salt has the optimum salt concentration is important. All right, that's a B. And third category, it also depends upon the temperature. So temperature is one such factor that also regulates turning on and off the enzymatic activity. And our body temperature is 37 degrees. So almost all of them, all enzymes perform efficiently at 37 degrees Celsius. If it goes higher, let's say 42, 45, then our body gets sick. We got the heat shock. So as you probably know, some of you, if you work in the military, when someone is transported from one region to another region, so cold to hot to cold, or vice versa, then basically body needs some time to adjust. You feel a little bit uneasy, and after a while it becomes okay, because body can tolerate certain temperature, right? We have some other proteins that can do it. And there are some other factors up there too. I'm not going to go there. And these are called others, all right? Those others are basically specific, the modification of proteins, binding of some small molecules, etc., etc. Not good, but you should know the factors that regulates the enzymatic activity: pH, salt, temperature. So I can ask the question, right? I can ask what is the definition of an enzyme? Enzyme has no structural change before the reaction, after the reaction. It lowers the activation energy and speed up the reaction. You must remember that. That's the characteristics of the enzymes. What are the different properties of enzymes? Enzymes have a catalytic power. What is the catalytic power? I can ask questions. Enzymatic specificity. What is the enzymatic specificity? I just showed you. One enzyme binds to only one type of substrate. Enzyme regulations. What are the factors regulates the enzymatic activities? A, pH, B, salt, C, temperature. Temp is temperature, and others. All those things are included in the enzymatic activities. Any questions? If not, then I'm going to the next one. So in our body, we have thousands of enzymes. Someone say open again. So we have thousands of enzymes. Does it mean that we have to study all of the enzymes, their characteristics? Answer is no. All those thousands of enzymes could be classified into 6 different groups. We call 6 types of enzymes. So all enzymes, thousands of enzymes, are in fact belong to only 6 types of enzymes. A type of enzyme, number 1, is called oxidoreductases. Remember the name of those electron transport chain enzymes? First one, what is called? NADH oxidoreductases. So that's basically does the oxidation reduction reaction. So oxidoreductases are they are the redox reaction. Redox reaction, redox reaction. Redox reaction is the type of reaction, two types of reaction, the oxidation and reduction. Oxidation is the addition of oxygen, reduction is the addition of hydrogen or removal of oxygen. So they perform the oxidation reduction reactions, right? Example, I already gave you before, and I'm going to put that on, is a ETC, electron transport chain. ETC. What's the ETC? Electron transport chain in the mitochondria. All those are redox. They are not the only redox oxidoreductases. There is thousands of oxidoreductases are also present in our body. I just give you one such example, ETC. That's good enough, right? Next one is called transferases. Transferases, remember the transferases? So transferases, they transfer something. What do they transfer? For example, let's say glucose. We have a GLC, right? And remember, once it enters inside the cell, what does it become? It becomes GLC 6P. 6P. So enzyme is hexokinase. Let's say enzyme is called hexokinase. Enzyme is hexo means 6, glucose has 6 carbon, hexokinase. So what does it do? Tell me, what does it do? Hexokinase, what is transferring? You have ATP. ATP is converted to ADP, diphosphate. Adenosine triphosphate is converted to adenosine diphosphate, and it's transferring the phosphate. It doesn't have a phosphate, now it's transferring the phosphate. So it is called transferases, one of the examples, and we have thousands of that, hundreds of that. Next one is the hydrolases. It does the hydrolysis reaction. So hydrolysis reaction means, let me give you this one. So this is the compound that you have seen it, right? This is an ester. Let's say this is the ester, right? You have a carboxylic acid and you have alcohol. And now the enzyme, the hydrolase, it takes the water H2O, all right? H2O. And enzyme is the hydrolase, all right? Enzyme is right here. It's called ester hydrolase. Ester hydrolase, because the compound is ester. So ester hydrolase, and it makes carboxylic acid, RCOOH plus R1OH, alcohol. So it is a ester hydrolase. It's hydrolyzing the ester. That's a hydrolysis reaction. Lyase basically, they basically cut. The lyase cut bonds. And I'll give you some example within a minute. Cut bonds. What type of bonds it cut? It can cut any type of bond. For example, let's say you have a C bound to the C. All right. So let's say you have like this. Carbon has 4 bonds, right? Remember? So you have a CC bond. And then remember, this one can cut into like this. C plus C. Can anyone give an example of this kind of a? So it's a carbon-carbon linkage. This cuts the carbon-carbon bond. Have you seen this one kind of reaction? Probably you did. Let me refresh your memory. It's aldolase. Remember the glycolysis? It's aldolase. Aldolase basically cuts the carbon-carbon bond. You can also have it can cut carbon-nitrogen bond, carbon-hydrogen bond, carbon-oxygen bond, carbon-sulfur bond, anything. Those kind of bonds, when they cut it, it's called carbon cutting the bond. Those are lyases. Isomerases. They convert the isomerases. Remember, we have for example, let's say we have a R, and let's say this is H, for example. Please write down R, H, C, double one, C, R1, and H. What type of configuration is this? What type of molecule is this? What type of molecule is this? You have it. Tell me. what is the configuration of this molecule? And it is changing to this H and R1. Come on, you already have it, you know. So this is a cis, both hydrogen on the same side, right? Cis, and this is the trans, cis and trans. And inside our cell, any reaction takes place, they must need enzyme. So this one is called cis trans isomerase, cis trans isomerase, cis trans isomerase, isomerase. So isomerase is a reaction, isomerase, all right? So cis trans isomerase converts the cis configuration to trans configuration. So this is a chemical reaction. Ligases basically adds bond. So basically add bonds. All right. So difference with the add bonds. All right. So ligases cut bonds, cut CUT, cut. Ligases cut bonds. All right. And ligases add bonds. For example, if you have a carbon compound plus, let's say we have a nitrogen, I'm giving general example, right? Now they can with this kind of ligases, they can add the like molecule like this, all right? N this. It can have anything. I just put N. So it's a carbon nitrogen bond is ligated by the ligase, So cut bond cuts it, ligases adds bond. They're basically reverse of the two things. All right. So that's kind of, these are the different types of enzymes. 6 types of enzyme. They carry out this type of reactions. Any questions? I will ask what are the, how many types of enzymes are there, and I can give you a specific example. Let's say this is the reaction done by this enzyme. What is the name of the enzyme? I mean, what type of enzyme is this? Is it oxidoreductases? Is it a transparases? It is a hydrolases? It is a lyase, isomerase, and ligase. Only 6 types are possible. Any questions?
I have a question about the hydrolases. What does the arrow indicate again?
So, this arrow. So this arrow is, it will be, name will be ester hydrolase.
Okay.
Ester hydrolase. Yeah, ester hydrolase. Yeah, you can ignore the arrow if you write like this. Ester hydrolase does the hydrolysis reactions. Any other questions? If not, so these are the enzymes, and enzymes have their different characteristics. All thousands of enzymes could be characterized or could be classified into 6 different groups: oxidoreductases family, transparases family, hydrolases family, lyases family, isomerases family, and ligases family. All right, only 6 types of enzymes are, whatever the enzymes we have, they perform, all those reactions could be classified into 6 different groups or types of enzymes. Any questions? If not, we're moving into it. Yes? All right. If not, then we are going to go to the cyclic and conjugated double bond, linear and cyclic conjugated double compounds with a conjugated double bonds. And those are basically known as heterocyclic compounds. There are 2 types, there are many types of heterocyclic compounds. We need to know only 2 of them, 2 different classes. One is called purines, other one is pyrimidines. Purines and pyrimidines. Purines and pyrimidines. Anyone has any question before we go to that? Yes? Hello? All right. If not,
Can you go back a slide really fast?
okay.
All right, thank you.
You're welcome. All right, I'm skipping something. Do not worry about it. We don't have much time to do all those things. So anyway, so heterocyclic compounds, remember that we talked about cyclic compounds, right? And among the cyclic compounds, if you have the hexo, right, cyclohexane, is the cyclohexane is most stable compared to cyclopentane, cyclobutane, cyclopropane, etc., right? We talked about that one before that. We also examined that one also. But I might ask that question again. So in cyclic compounds, what is the order of stability? So cyclohexanes have the greatest stability than compared to cyclopentane, cyclobutane, and cyclopropane is the least stable cyclic compound. So what are the heterocyclic compounds? Heterocyclic compounds are basically they have a hetero, meaning they could have a nitrogen, carbon, hydrogen, carbon, sulfur, oxygen, etc. Examples. So heterocyclic compounds, there are many heterocyclic compounds are there. One family is called purine family, other one is called pyrimidine family. All right. So the purine family, the skeleton of the purines looks like this. So let's say purine. All right. It is a basic structure, and that basic structure looks like this. Please write down with me. It's a 6-membered ring. You have a nitrogen here and nitrogen there. So far so good. And then let's say it has a double bond like this, double bond, single bond, double bond, and double bond. All right. So then this one is, I'm sorry, I'm rewriting it again. Please pardon me. I'm going to write a little bit different. It is not the because I have to put another this side. So hang in there. So let me write down this. All right. So you have a N and N. I'm just flipping it because I'll tell you why. And then there's another ring looks like this. Like this. So it's a hexa, right? 6-membered cyclic. It's a cyclohexane type of molecule, but it is a nitrogen in there. And there is another nitrogen here, another nitrogen there. So this is N. Now put the double bond. All right. So double bond, and double bond. Right there. All right. And NH. There's H here. So this is called purine family. And why is called heterocyclic? Because it has the hetero, it is a carbon, hydrogen, nitrogen. That's called a heterocyclic compound. Purine family of, and there are many derivatives up there, and I'll show you some derivatives very soon, okay? And next one is a pyrimidine. So let's say pyrimidine, how the pyrimidine, the skeleton looks like. They are heterocyclic compound, right? So pyrimidine.
I have a question.
Yeah.
Is the second molecule in the purines, is that only have 5 sides?
That's right. So it is a
Okay.
it is a 6-membered ring plus 5-membered ring together. Yeah. That's the heterocyclic and compound. Heterocyclic. So two cyclic molecules are together. That is correct. Purines, the pyrimidine family.
1150.
Is already done?
Yeah.
All right, I'll start from Wednesday.